All right, so yes, we want your samples, but we definitely have some expectations. There are um just a few things that are going to be necessary to keep in mind when you're going out in the field and collecting mushrooms and taking pictures of things. The most important thing is going to be proper field documentation, and that's going to be um as simple or as complex as you want to make it. But I guess like a holotype collection that you could see right here. It's got a nice underside photo of the gills. You can see different growth stages of the mushrooms. You can see the environment. That is the most important thing we need right now, because GenBank is the place where all the secessions for sequences are, and people don't have location information, photos, notes, anything like that. So anytime we can have those types of observations to our posters, just make them miles above the rest. The way we do that to preserve them after we find it is going to be through just a food dehydrator. And then we're going to put them in just little collection bags. This is just kind of a quick little overview. But I guess the best way to document your photos is going to be if you're walking out in the woods, you stumble across a nice little patch of amanitas. And the first thing we're going to want to do is take a picture of them in their undisturbed original habitat. This is going to show us the substrate, which in this case, they are growing mycorrhizally from the ground. We could see that there's also pine needles there. This specific is Pinus taedae, the loblolly pine. So, a photo can speak a lot of very valuable information. Such is true with amanitas. You need to be able to dig them all the way up. They have vulvas. Entomopathinogens often have mushrooms that are attached to bugs, and those are key important identifiers and just great things to observe moving forward. You definitely want to be able to note the varying maturity. You can see that some of the caps have opened all the way, some of the caps have not broken their veils yet, which also goes to show that the underside is going to be one of the most important parts and the often most forgotten aspect to photography. It's important to note the gills, the way they attach to the stem. They lots of different formations for that. There's different spacings for your gills, and being able to see all of that is very important. Also, cutting and damaging the mushroom, as you can see with many boletes, is if you pluck them, you can see on the bottom here where my finger was, that leaves a little bit of a blue imprint. And also when the inside of the flesh is exposed to air, it gets very blue. So that is also important signifying identifiers. Gills of lactarius bleed different latex. Certain mycenas have like a bloody liquid that comes out of them, and sometimes they change color in time, sometimes they don't. It's also very important to note those. And not necessary but great bonus is microscopy. This is I think it was an Asco corne that Annie took a picture of. This is just a beautiful photo. Yeah, microscopy, I think, is a very underrated aspect to fungi documentation. A little bit further, this is what, I don't know why it's so blurry, but this is just kind of all the important aspects and all the valuable parts to an iNaturalist observation. We're going to see that we have a great picture of the mushroom and its environment. We have a photo with KOH on the cap. That's going to be the orange little dot. We have some strikes across the mushroom to show the bruising. I have some notes that it was growing with various Quercus species and oh my God, the American beech. And you can see that it's got a location. It has the time you observed it, the day you submitted it. I can run through a quick iNet upload, I guess, for folks. I'm not sure if folks are familiar with that. It can be a little daunting at first. I'll go very quickly. So it's much easier on the phone, on the app, because it's just one button and it asks you, it'll be an immediate pop-up that you can either take a photo or choose from your photo library. And it's going to be essentially the same thing we do here. So we go ahead and we have our picture of our mushroom that we can add to iNat, and the AI, the artificial intelligence here at iNat is gonna, I guess, give us a suggestion, but it's gonna take a little bit because I guess I'm on Zoom here. But you're able to put the date in, you're able to put the genus and what location, and you just find a, I could say like Wood End Nature Sanctuary. This isn't from Wood End, but just for the sake of being. So now that we have an accurate location, you'd be able to say that the mushroom stained, yada, yada. So make your observation. And then the most important thing is it's going to give you a number. And that number is then your, I guess, the session for that specific observation. And let's try to show a quick example of that. Let's go to my most recent one. So there's going to be a 9 number at the end of your observation. And what we use these numbers for is our field data slips. And that's going to be a way that we can keep our documentations in order. As you can see, this is just one container from my last weekend in North Carolina, and a lot of samples pile up very quickly. And the best way to take care of that is if you have that iNaturalist number, and even if you have chicken scratch handwriting like me and you can't read half of the stuff that I wrote here, there's an iNat number that will be the most valuable aspect for moving forward. These slips also, they help credit identifiers, the collectors, the habitat, any notes or other observational features that you might have forgot to put on the computer that you can just scribble down when you're out in the field. It's just very important. It helps us, as you can see, like once you start piling samples up in the dehydrator, there could be 20, 30 samples at a clip and even more. So you need to be able to know which one is which. And once mushrooms dry out too, they look very different than when they did when they were fully hydrated. But the most important thing when you're dehydrating is that you keep your dehydrator at the lowest temperature as possible. It's going to be between 95 and 105 is often the lowest setting. That's due to the fact that too high of a temperature can start to damage the DNA that's present in the mushrooms, and that's exactly what we want. 24 hours is about the gold standard for a mushroom. Some like Suillus take, or like larger Cortinarius, Amanitas can take a lot longer, but you can generally tell they have a very dry, crackery consistency to them. And yeah, once they're fully dry and that you have that cracker consistency, you want to make sure that they're placed in something like a Ziploc bag. That's going to make sure that no more air can get in, that there's going to be no more hydration. They'll stay relatively nice and fresh for sequencing in the future. You can also, like you saw earlier, put it in a container when you're finished to keep a little bit more air out. And it's optional, but not necessary. I think that's going to be it. Yeah, just obviously dropping by one of the meetings when we have them is going to be the easiest way to get them to us, or like just contacting one of the group members. I'm not sure who's speaking next. It's probably Annie or Megan.
Yep, it's me, Zay.
Cool. I will go ahead and pass it to Annie then.
Thank you. That was great. So important to do all the things they said, because as someone who has way too many mushrooms, there are many times that I have not done the right documentation. I've had to toss them out, and that's really sad.